Good morning, everyone. I am Ratthakorn Sumetcherngpratya, and today I want to talk about my research entitled Expression of Endothelin-1 and Endothelin receptor in human periodontal ligament cells from adolescents and adults. Nowadays, adult patient has high seek to orthodontic treatment. And one of the challenges in adult orthodontic treatment is the slower rate of tooth movement than adolescents. The differences in biological response between young and adult patients are one of the factors determining the rate of tooth movement. Force is needed to apply in order to move the tooth. When force is applied to a tooth, the pattern of cellular activity and blood flow in periodontal ligament, as well as many mediator and second messenger, such as adenosine monophosphate, interleukin-1 beta, interleukin-6, which is the pro-inflammatory cytokines, are changed. Additionally, apart from that changes, continuous force also upregulate the endothelin-1 expression in PDL's microvasculature. Previous studies show the upregulation of endothelin-1 immunoleveling in the endothelium of the rodent PDL. Endothelin-1 is the potent vasoconstrictor peptide consisting of 21 amino acids. Endothelin are produced in various cells such as endothelial cells, fibroblasts, smooth muscle cells, epithelial cells, and also in PDL cells. One of the endothelin-1 function is the stimulation, the synthesis pro-inflammatory cytokines, such as interleukin-1 beta, interleukin-6, and TNF-alpha in PDL cells, involving the bone resorption and tooth movement. Endothelin-1 action is evoked via its receptor, endothelin receptor A and B. Previous research has demonstrated that specific receptor antagonists accelerated or deaccelerated the rate of tooth movement in mice. For example, endothelin dual receptor antagonists enhance tooth movement, while selective endothelin A antagonists slow the rate of tooth movement. Therefore, endothelin and its receptor may play a role in the rate of tooth movement. However, there is little information about the expression level of endothelin and its receptor between adolescent and adult. So we hypothesize that the messenger RNA or mRNA expression of endothelin-1 and its receptor in human PDL cells may change with age and contribute to the rate of tooth movement. So the aim of this study was to compare the mRNA expression of endothelin-1, endothelin A and B receptor in human periodontal ligament cells from adolescents and adults. In our method, PDL cells were obtained from non-carious bicuspid teeth with healthy periodontium that were extracted for orthodontic reasons from 5 healthy subjects without non-parafunctional habit in each group. In adolescent group, patient age was ranged from 14 to 20 years old. Mean age was 50.6 years old. And in adult group, patient age was 30 years or more. Mean age was 32.6 years old. The sample size was calculated according to the previous study with 80% probability function power and a 5% significant level. PDL cells were obtained exclusively from the middle portion of the tooth root and placed in the tissue culture dishes. After conference in 100 millimeters tissue culture dish, PDL cells were passed in which between third and second passages were used in the experiment. Then RNA was isolated using the total RNA mini kit. And 2 micrograms of total RNA was reverse transcribed to single-strand cDNA. Then endothelin-1 and its receptor expression were accessed by real-time PCR or qPCR. These are the primer sequences that use in the experiments, we use the GAPDH as the housekeeping gene to normalize the gene of interest before comparing between groups. One sample from the adult group was designated as a reference, and the relative gene expression of other sample were calculated as a fold chain of the reference sample using the double delta CT method. The experiment was performed in triplicate and repeated twice. Independent t-test was used to evaluate the differences of endothelin 1, endothelin A and B receptor mRNA level between adolescents and adults. And the level of statistically significance was set at p-value less than 0.05. Let me show what we find out in our research. This figure shows that the relative mRNA expression of endothelin 1 in adolescents was significantly higher than adults. And similarly, the expression of endothelin A and B receptor expression in adolescents was also significantly higher than adults. Endothelin 1 occurs through its receptor A and B. Endothelin receptors are expressed in many organs, including periodontal tissue. Previous studies showed that endothelin A and B receptors are weakly expressed in PDL cells by Northern blot analysis. However, using this experiment qPCR, our study has confirmed that the endothelin 1 and its receptor mRNA are expressed in culture PDL cells from both adult and adolescents. The previous study demonstrated that interleukin-1 beta, interleukin-6, and TNF-alpha mRNA and protein level in both healthy and periodontitis PDL cells were elevated by endothelin 1 in dose and time-dependent manners. This pro-inflammatory cytokine regulate the inflammatory response and bone remodeling process, which is the key in orthodontic tooth movement. Our study demonstrated that endothelin 1 mRNA expression was significantly greater in adolescent than adult, which might affect the pro-inflammatory cytokine level and resulting in the different rate in the tooth movement. One of the main action of endothelin A receptor is stimulation of pro-inflammatory mediator secretion. As mentioned before, previous report demonstrate that the selective endothelin A antagonist slow the rate of tooth movement by increased alveolar bone volume and decreased osteoclast activity. Therefore, it would be possible that the higher endothelin A receptor mRNA expression in the adolescents, the more pro-inflammatory mediator will produce and causing the faster tooth movement. And however, further study is still needed. On the other hand, one of the main action of the endothelin B receptor is endothelin 1 clearance. Previous studies show that endothelin B receptor knockout mice show increased endothelin 1 level, decreased osteogenesis, and impair osteoclast activity, resulting in decreased amount of orthodontic tooth movement. Moreover, the high endothelin 1 level result in a significant reduced density of endothelin A receptor, which might be correlated with decreased pro-inflammatory mediator secretion. Our study showed that it would be possible that the higher endothelin B receptor mRNA expression level in the adolescents may cause the faster tooth movement. Even though the endothelin A and B receptor involved in bone remodeling and contribute to regulating the cytokine secretion, little information is known about the downstream consequence of endothelin A and B activation in PDL cells. So additional investigation has post-transcriptional level, and the effects of endothelin-1 treatment on PDL cells, such as pro-inflammatory cytokine production, needs to be assessed between adolescents and adults. In summary, our study demonstrates that the level of mRNA expression of endothelin-1, endothelin A and B receptor in PDL cells from adolescents were significantly higher than adults. So this result suggests the possibility that the endothelin system may play a role in the different rate of tooth movement in adolescents and adults. Thank you for your attention. Does anyone has any question?
Now we're going to wait for the question from the audience. Okay. Okay, Captain, going to be me first. All right, from your experiment, from the tools you select, why do you think that the movements of the patients less than 24 years old and over is different? Because you use the tooth group with 14 to 20 and the other group is 30 years old. What do you think that both groups have movement different?
Because in my review literature, the mean cutoff age for adolescent group is below 20 years old, and my oldest person in the adolescent group was 19.
Yeah, my question is because you want to test like ET1, right?
Yes.
Which related with the movement of the tooth.
Yes.
So I'm just wondering why you think the two groups have the tooth movement different?
Related to endothelin-1 and age, right?
Yes. Why you think this your selected group might have different level?
They have no previous study research, but in the clinical situation, the patient who not growth, which is more 20 years old, have the different rates of tooth movement. So we use the two group of age that the mean cutoff age is 20 years old.
Mm-hmm. And do you think that this is tooth movement? Do you think that extraction force before extract the tooth will have effect on the level of your RNA expression?
Maybe affect this expression. But when I collect the tooth sample from the surgery department, I observe the doctor and ask him or her to do it gently.
Okay, so they all both experienced doctor and do it very fast and gentle, huh?
Yes.
Okay, so from your study, why do you think that the level of the adult is less than adolescents? And lower, right?
Yes, but why, right?
Why it's such a significant difference?
Maybe because of the action of the endothelin-1 action and their receptor, because their action is about the secretes the pro-inflammatory cytokine and enhance bone remodeling So it is the correspond that in the adult group, which is have the slow rate of tooth movement, will have the low gene expression in this gene endothelin 1 and its receptor.